Today's topic is about comparison of different blotting technique. Mainly three blotting technique are utilized: southern, northern and western. First we will look about the pictures of these blotting technique, then you have to say which blotting technique is there. So here first DNA fragments that is separated with the help of that gel electrophoresis. Now initially the bands not visible. Now that bands whatever the generated over this electrophoresis that will be transferred to the paper. Now that transferring that is called blotting technique. Once that blotting is over, then add a particular probe to the visualize the band. Now this probe which is utilized in this technique, that is particular DNA. So DNA probe is added, and if that particular complementary sequence over the particular probe blotting paper is present, then it will be visualized with the help of this autoradiography. Otherwise there is no band will be seen in the particular autoradiograph. And this technique to detect particular fragment of the DNA or particular part of DNA or the gene, that is called a southern blotting technique, because it is detecting the DNA detection with the help of southern blotting technique. Next here that RNA is separated over the gel electrophoresis. Again that not visible band, but that will be visualized with the help of adding of a probe. Here which type of probe is added? That is complementary DNA. This complementary DNA is generally produced as a complementary to the mRNA sequence with the help of reverse transcriptase enzyme. So here again that DNA, particularly complementary DNA probe is added. Now again that probe that is helping to visualize the band if particular complementary sequence of the mRNA or the RNA is present with the help of autoradiography. So this technique is called a northern blotting technique. Last one, that is protein detection. So this protein is again electrophoresis. It is also not visualized under normal, but when it is added with a particular probe, here that probe that is generally utilized as an antibody. So antibody will detect particular antigen considering this protein, and this particular antigen or protein is present, then it will be visualized under the autoradiography. So this protein detection method that is called western blotting technique. So simply you have to remember that DNA detection with the help of southern blotting, RNA detection with the help of northern blotting, and protein detection with the help of western blotting technique. Yet DNA probe is utilized in a southern blot, complementary DNA probe is utilized in a northern blot, and particular antibody, radio labeled antibody is utilized for the western blot technique. So that is comparison and some similarity between different blotting techniques.